Our next speaker is going to be Jonathan Lunine from the Jet Propulsion Laboratory and Caltech. And Jonathan will talk about ocean worlds and habitability.
Okay, thank you, Matthew. So ocean worlds are bodies that have substantial amounts of liquid on their surfaces or within their interiors, and usually we think of liquid water, although in the case of Saturn's moon Titan, there are substantial stable seas of liquid methane and ethane. You can make an argument about whether Io counts as an ocean world with its lava lakes, but I would say no. These are very interesting objects from the point of view of the potential for studying oceanography off of the Earth, for the origin of these oceans, and for habitability. And I was told that I was going to talk about habitability, so that's what I'm going to talk about. So here are all of the places that have been implicated as potential ocean worlds. Of course, the Earth has ample surface water. There are places that probably had liquid water in their past. And then there are these places that are either suspected to have or essentially known to have liquid water under their surfaces. And the essentially known to have are Europa, Enceladus, and Titan. Europa and Enceladus in particular, there are multiple lines of evidence for liquid water under their surfaces, and in the case of Enceladus, it's actually possible to study the composition of that material. So what one wants to understand is whether these liquid water environments are habitable. Can they sustain life? There is a lot of discussion about what the term habitability actually means. There's a very nice article by Charles Cockle and colleagues in Astrobiology from 2016, and I recommend that because it has an almost encyclopedic discussion of the question of habitability. There's instantaneous habitability, which is if some ice on Mars melts for 10 or 15 minutes, you throw a bacterium in there, the bacterium will survive. But really we're talking about what Charles and colleagues call continuous planetary habitability. That is the ability of a body to sustain liquid water over geologic time and over a large volume. So habitability, the ability to sustain life, requires some very basic properties, which are shown here. There is the requirement of some kind of usable energy, which could be sunlight that's converted through an excited electron to other forms of usable energy, and then carbohydrates. Could be chemical energy, some form of usable free energy. There's the need also for some kind of a solvent. Here we're going to be talking principally about liquid water. There are a lot of very interesting ideas about the possibility that liquid methane with ethane might be able to sustain a kind of biochemistry. There was even a KISS workshop on that some years ago. But since that time, the excitement, I think, has moved back to liquid water because we know so much about the ocean of Enceladus, and we will learn a lot about the ocean of Europa in the coming years. And then, of course, one needs the elements of life. Now, life uses essentially all of the elements that are available to it in its environment, but the fundamental ones, the core ones, are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, which play seminal roles in Earth life. And then the appropriate physicochemical conditions, that is temperature, pH, and so forth, in order to maintain biological structures, polymers, membranes, and so forth. Now, the conditions under which the building blocks of life can be constructed are probably much broader than the conditions under which known life can exist or be active. And on a temporal sequence here, and this is from Cockle et al.'s paper in 2016, between the environments, the astrophysical and planetary environments where the building blocks of life, amino acids, nucleic acid bases, and so forth, are produced, and habitable environments which can sustain life, there is a bottleneck, and that bottleneck is the formation of life itself from non-living chemistry. Those conditions are not at all understood because the mechanisms by which life comes from non-life, abiogenesis, is still a mystery after a century of work on this topic. But plausibly, that set of conditions is smaller than the set of conditions in which life can be sustained. And in fact, as life evolves on the Earth, we have examples of organisms that have become capable of existing in very exotic environments. So this area, this patch, grows with time. Because of that bottleneck, though, one should be prepared to find environments that are habitable, that can sustain life, in which there is no life at all. And finding that that is the case, for example, in the oceans, the outer solar system, the subsurface ocean of Titan, of Enceladus, of Europa, that would be very interesting. Now, as I mentioned, life on Earth as it evolves has produced organisms that are capable of existing in very extreme environments. And so habitability is to some extent dependent on the particular organisms you're talking about. So in this figure from Schock and Holland, temperature is on the y-axis, pH is on the x-axis. Life that is familiar to us, including us, tends to be in this green box that you see here, familiar life. However, there are life forms that can exist, for example, in pHs that are equivalent almost to that of household bleach. There are other organisms that can exist in a liquid that is more acidic than battery acid. There are organisms that can exist as long as they have access to some amount of liquid water in the form of brines that can exist well below the freezing point of water. And there are organisms that exist well above the 1 atmosphere boiling point, such as this one here. And so in all of these cases, these are life forms like us with DNA, RNA, proteins, and so on, but they have evolved particular survival strategies, particular proteins, particular high rates of DNA repair in the case of radiation-tolerant organisms, and therefore are able to exist in a broader range of habitability space. Whether primitive organisms, if you were to go back to the earliest part of the habitable history of the Earth, whether those organisms, the earliest, would have had the same sort of capabilities, or whether habitability would have been restricted to a much smaller space, parameter space, remains an open issue. Now, I'll talk about Enceladus in a moment. In terms of planetary habitability, there's really one place in the solar system, beyond the Earth, that we know enough about that we can say is a habitable liquid water environment, and that is the ocean of Enceladus. And so this is a table modified actually from Cockle et al., updated. The molecules that have been found in the ocean of Enceladus include both carbon monoxide and carbon dioxide on one end of the redox scale for carbon, and methane and organics on the other. Hydrogen in various forms, nitrogen in various forms, and most recently, in a paper last year looking at Cassini data, Postberg et al. Found phosphorus in the form of phosphates. Phosphorus is very important for life. Phosphate bonds tie together the sugar backbone of DNA and RNA, and the phosphate bond is the energy currency for life through ATP and ADP, adenosine di and triphosphate. And then there are some potential energy sources in terms of redox reactions here, which I'll talk about a little bit later. So what are the key types of measurements required to establish habitability? Well, those for planetary environment, such as we will talk about, are quite varied. They're geophysical, they're chemical, they're isotopic, and they're also electromagnetic. The ocean of Europa was discovered by the magnetometer on the Galileo orbiter by detecting the induced magnetic fields that were present around Europa caused by the time variable Jovian magnetic field passing through what was evidently a relatively high conductivity region in the interior of Europa, but not very deep, relatively near surface. And through several flybys of Europa, it was possible to establish that this must be a liquid water ocean. So a number of different criteria for habitability are shown here and their relationships: composition, pH, salinity, the distance from equilibrium for different kinds of metabolisms, potential metabolisms, the activity of water relative to pure distilled water, temperature, density, thickness, pressure, circulation, and the longevity of the ocean. One of the questions for the ocean of Enceladus is whether it has been around long enough or continuously enough to have had life actually begin and then to evolve. And determining the longevity of an ocean inside an ocean world is a very challenging proposition. So these different parameters are coupled to each other. Here's an example for Enceladus, could be also for Europa. But there are three parameters associated with habitability that are plotted here. There's the pH on one axis. There's the redox state in terms of carbon on the other, the x-axis, if you will, and then on the z-axis here, free energy for metabolism. The habitability space, the goodness of that environment for supporting life, is illustrated by these contours. Dark green is very supportive of life, light green less so, and then the grays essentially marginally habitable or uninhabitable. This is all qualitative. But as you can see, depending upon where you are on the free energy axis, the habitability space is larger or smaller. If the free energy available is very small, that limits the kinds of metabolisms that organisms can use to survive, and that in turn might limit the both redox and pH space that these organisms can exist in, or their temperature space if you put a fourth axis on here. So understanding all of these through measurements at these ocean environments themselves is very important in understanding the habitability of these environments, whether or not you detect life. So let me talk concretely now and start with Enceladus. Saturn's moon Enceladus has been the easiest to explore in terms of its subsurface ocean, and the most is known about it thanks to the fact that the Cassini orbiter, which was at Saturn from 2004 to 2017, made multiple flybys of Enceladus and 7 fly-throughs of its plume. First of all, Enceladus is a very small moon of Saturn. Here it is compared to the Earth's moon. Europa, which we'll talk about a bit later, is just about the size of the Earth's moon. So it's somewhat of a surprise that Enceladus is so active, and we presume that this activity is due to tidal heating, even though Enceladus is not currently in a precise resonance with any of the other moons. It's in a near resonance with one of the other moons of Saturn. But Cassini discovered early on this plume of material coming off of the south polar region of Enceladus, a plume of gas, dust, and ice. It goes into orbit around Saturn because of the low gravity of Enceladus to form what's called the E-ring. Some of the mass spectra you'll see in a minute are from the E-ring, some are from the plume itself. The plume is fed by these jets near the surface that correspond to fractures. Here are the main fractures here, but these are on subsidiary fractures, and the source of that material is believed to be an ocean, and there are multiple lines of evidence for an ocean, although not from magnetic measurements, from other measurements I'll describe in a moment. What you'll see in a minute is mass spectrometry data. Mass spectrometers separate species by mass. Species are charged, first of all, they're ionized, and in the case of Cassini, that ionization is actually due to collisions with the instrument itself as Cassini moves through the plume. There are 2 mass spectrometers. One is a quadrupole mass spectrometer in which you have 4 conducting cylinders to which electric fields are applied that are time variable, and by tuning those electric fields, you can move ions of different masses through this to the detector, through the cylinders to the detector, and thereby scan the different masses. Time of flight mass spectrometer is the other type of mass spectrometer on Cassini. Here the ions are accelerated into a chamber. An electromagnetic reflectron is used to reverse their course. As they move through here, the different velocities that they get because of their different masses allow them to be separated spatially and therefore to hit the detector at different times. And that allows then for the mass to be determined for these ions. So first and foremost, the mass spectrometer showed that the icy material coming out of Enceladus is salty, at least the largest mass grains. So the time of flight mass spectrometer has a collision plate where the ionization occurs. That collision plate also determines the mass of the grains because the velocity is fixed by the spacecraft velocity. So the very largest ice grains turn out to be the saltiest. Here's a mass spectrum, log of amplitude versus mass in atomic mass units. You see potassium, sodium, chlorine, and CO3 as well. So various components of salts. And the salt content is between 0.5% and 2% relative to water, which is too high for dissolution in water ice. And therefore, these ice grains almost certainly were originally liquid water, which when they left Enceladus were flash frozen and the ice was trapped. So that's one line of evidence for liquid water, but that the liquid water is in a subsurface ocean came from two other lines of evidence. One is that Cassini measured the mass distribution inside of Enceladus through gravity science, measuring the gravitational field, fitting those data to a model which includes rotation, which was what McKinnon did in 2015 in GRL, showed that underneath the ice crust of Enceladus and above the silicate layer, and Enceladus's density is high enough that most of the mass of this moon is actually silicate, there must be a region that's slightly denser than water ice and is global in extent and is inferred to be liquid water. The fact that it is liquid comes from a different line of evidence. Imaging of Enceladus, again by Cassini, shows a libration, a rocking motion associated with the eccentric orbit of Enceladus around Saturn. The amplitude of that libration, the angular amplitude of that libration is several times larger than what you would expect for a solid body of the mass of Enceladus orbiting around Saturn. So the explanation, given in the paper by Peter Thomas et al. And Icarus, is that the crust of Enceladus is decoupled from the deep interior by a frictionless layer and is therefore being torqued to a larger extent. The amplitude of the libration due to the torquing is much larger than for the body as a whole. And so that implies, again, a liquid layer underneath that ice crust and above the silicates. So those two together are very strong evidence for an ocean, even though we don't have induced magnetic field data and cannot have it because Saturn's magnetic field is so quiescent. And then finally, the fractures themselves are much hotter than the surface. Surface is 70 Kelvin. The fractures radiate at 200 Kelvin. They're probably hotter even deeper down. That's a huge temperature difference consistent with an ocean being underneath. So what else is in that ocean? Well, Cassini's mass spectrometers measured organics. This is again from the time of flight mass spectrometer, the CDA. This is relative amplitude. Here now plotted is time of flight, but the mass numbers, the atomic mass unit numbers are given here at the peaks. And you see this collection here of peaks that are separated by 12 or 13 mass units, which is carbon or carbon and hydrogen. These are identified to be high molecular weight organic compounds because they are separated by a carbon or a carbon and hydrogen. But because the mass resolution of the Cassini mass spectrometer is poor, we don't know what their identity is. And then Again, in a paper last year, a small fraction of the grains analyzed by Cassini from the E-ring show evidence of phosphorus in the form of phosphate compounds. Again, this is a small fraction, but the fact that phosphorus is present at all is significant. Now, the compounds for life are present, but is there the kind of activity needed to provide temperature gradients, redox gradients, and so on? The evidence seems to be yes. Here's another example of CDA spectra. Here you see, again, amplitude versus time of flight. You see silicon and oxygen in a 1:2 ratio, so that's silica grains. They're very small. Their mass distribution is very narrow. They're almost monodisperse. And so they are material that has not spent much time in that ocean. Otherwise, it would have grown to have a broader mass distribution. They're injected into the ocean from the silicate region and ejected into space relatively quickly. So where does the silica come from? Well, it potentially comes from hydration reactions. Here's one example. Phthalate and water forming magnetite and aqueous silica, and the excess hydrogen should be present as molecular hydrogen. The other mass spectrometer on Cassini, the INMS, detected molecular hydrogen on the very last fly-through of the plume. And so this all hangs together as indicating that there's active water rock chemistry going on at the base of the ocean. And there's even evidence that that ocean has redox energy available to it. If we postulate that the carbon dioxide, the methane, and the hydrogen, all detected by the INMS, are related by this reaction, which in biology would be methanogenesis, given their abundances, we can calculate what the redox state, what essentially the Gibbs free energy of that ocean is relative to this reaction. And so that's what's plotted here. This is the chemical affinity, which is effectively the difference in the Gibbs free energy from equilibrium versus the hydrogen mixing ratio. The pH is involved here because while the ratios of CO2 and methane and H2 are probably preserved as they're ejected, their ratios relative to water are not. So their ratios, their concentration in the water has to be determined by a more complicated calculation which involves pH, and that's why you see that here. The result of that calculation done by Chris Klein and published in a paper by Wade et al. Gives this blue region for Enceladus, and that's well above equilibrium, and it actually overlaps with some of the environments in which methanogenic Earth microbes are present. So this is a reasonable departure from equilibrium for an aqueous environment where methanogenesis either is occurring or could occur. So what's next for Enceladus? Well, the low mass resolution of the mass spectrometers leads to a lot of ambiguities. Here are mass numbers and the species that could possibly have resulted in the peaks seen in the INMS data. But the Cassini mass spectrometer is not state-of-the-art, and being launched in particular to Europa next month is a mass spectrometer called MassSpecs, which has 80 times, that is nearly 2 orders of magnitude, the resolution that the Cassini mass spectrometers do. So for example, at mass 28, where you can't distinguish between CO and N2 and methane with the Cassini mass spectrometer, you can easily do that with the MassSpecs mass spectrometer, because again, the nuclear binding energies are different for these different elements, so they have fractional masses in addition to their unitary masses. And of course, a modern mass spectrometer would give you a much larger range of mass, and so going back to Enceladus with a mass spectrometer like MassSpecs and others would potentially be able to identify for us the presence of biological molecules at Enceladus. So very quickly now for Europa Clipper, here's a beauty shot of Europa Clipper with one of its solar panels unfolded at the Cape. It's a mission that is designed to determine the habitability of the ocean of Europa, even though Europa's ocean was discovered well before Cassini, well before Enceladus's. We know very little about it. Galileo did not contain mass spectrometers. There were no evident plumes. But the story for Europa, the story for Jupiter's moon Europa is that there is likely to be a source of redox energy through the production of highly oxidized materials in the radiation environment near surface. If those materials can make their way into the ocean, and there's chemistry at the base of the ocean where the silicate layer is, Europa, also like Enceladus, is mostly silicate, then there will be reducing compounds at the base, highly oxidized compounds at the surface, and a potential for a large amount of redox energy. The instruments on Europa Clipper will be able to tell us a lot about this ocean material if it finds its way to the surface. And evidence from HST and JWST data, mostly from Samantha Trumbo and Mike Brown, and also from the Goddard group by Villanueva, suggests that non-ice compounds are concentrated in the most youthful parts of Europa's surface. So we do expect to see material from the deep interior. The mass spectrometer is shown in red, remote sensing instruments from UV to visible to near-infrared to mid-infrared are shown here, a radar that may probe all the way through the crust, and then a magnetometer and gravity to probe both the salinity of the ocean and the potential topography of the ocean floor. And so if you map Europa Clipper's instruments onto these habitability parameters, you find that this mission, which by the way will not orbit Europa, it will instead orbit Jupiter and make up to 50 flybys of Europa, will be able to tell us a lot about all of these different habitability parameters. So let me close with 2 slides about the question of sample return. I've talked about in situ studies. Interestingly, ocean worlds sit in between Mars and exoplanets with respect to sample return and in situ studies. To understand the habitability of ancient Mars requires bringing samples back to the Earth for a wide variety of reasons, but Mike told us some of those in his talk today. For exoplanets, neither sample return nor in situ analysis are possible to study habitability. Only remote sensing can do that, and that's a very challenging task. For the ocean worlds of our solar system, remote sensing and in situ investigations, like in the plume of Enceladus, tell us a lot about the habitability of these oceans. And so the question is, do we really need sample return for those objects? So this is my last slide. Here's the pro and the con. Sample return will be good for moons that have perhaps relict oceans or for which the signatures of those oceans are very faint. Also, if one can acquire silicate samples, samples of the rocky portion, getting isotopes there will tell us a lot about the history, but it's not clear that's so important for Enceladus or Europa. There's the daunting problem that a lot of what one wants to do at places like Enceladus with very high resolution mass spectrometry requires such large data volumes that it may be impractical to return the data to the Earth, so maybe return the samples. And then most importantly, if you want to study cells, if you just calculate the amount of water that Cassini collected as it flew through the plume of Enceladus on one of its flybys, and you take the amount of cells per unit per volume in seawater on the Earth as a standard, what you find is that Cassini collected at most 1 or a few cells during each of its flybys, which is too small for a separate analysis unless you bring that material back to the Earth. If you want to do anything like genomic sequencing, you'd have to do that. Now, there are some cons here, and we can talk about these at length during the week, but very quickly, if you want to land and collect material, you then have a huge delta V that you have to exert to get back to the Earth from deep in the well of Saturn or the well of Jupiter. If instead you collect, let's say for Enceladus, material in a high-speed plume fly-through from solar orbit, those velocities are so high that you would destroy essentially all of the information in the organics that are present. Cryogenic sample preservation is really challenging, and most severe is the backward planetary protection requirement. Any mission that is going to purport to return cellular material that might be viable from the rest of the solar system to the Earth is going to have huge planetary protection problems, as we've discovered with Mars sample return. With that, I will stop. Thank you.